Okay, now that we learned about codons and anticodons and tRNA and ribosomes and all of those stuff, we can now finally move on to the last stage, translation. So there are three stages, but there are no names like that for transcriptions. Okay, so first, the exposed codon in A site attracts a complementary tRNA which bears an amino acid. So just look at this picture, okay? Trust me, look at this picture. This is A side, this is P side, this is E side. So, what first happens is that this tRNA, it first goes to A site, the first site that it has to go to. And so the codon, it attracts with the anticodon and causes the tRNA to bind to the mRNA, which is in the ribosome. So that tRNA is carrying, transporting the amino acid, and it goes and it brings it very close to it, and it brings it close to the next tRNA molecule, which is in the site that is next to it. So next, the ribosome forms a peptide bond between the two amino acids carried by the tRNA molecules. So as you see here, this tRNA came from the cytoplasm, bringing this amino acid, and it landed here. And there's also a tRNA here that is holding an amino acid. So what it does is that this ribosome, so since these two have a amino acid at the top, this ribosome actually forms a bond between these two amino acids in order for them to attach together. And this bond is called a peptide bond. That's step two. But first, but after they create the peptide bond, it breaks the bond between this tRNA and the amino acid so that the amino acid can go freely. If you see here, after this bond is created, this amino acid will come off of the tRNA. And yeah, that's happened. So the third one is that ribosome pulls the mRNA strand by the length of one codon. So this is the length of one codon, right? And so this ribosome, it pulls this strand. It makes it move this way to the left, right? By the length of one codon, which is like one, two, three. This three is one codon. So by pulling it to the left, by one codon, it will cause all of this will shift to the left one codon, which is like three of these units here, right? So this tRNA, which was in the E site, will now leave the ribosome and go to the cytoplasm. This tRNA in the P site will go to the E site, and this will go to here. And a new tRNA will be able to fill in this place that is empty now in A site. So by this mRNA shifting, to the left by one codon, the tRNA one will leave, the two others will shift, and it will provide another space in order for another tRNA to come and the amino acid chain to lengthen. So now this part will be exposed. The A site is now empty, and it exposes this part, which is the next mRNA codon, and it allows for the tRNA anticodon and the mRNA codon to be binded together. And so this process is continuous. It's like a cycle. It keeps on repeating until there's a stop codon. So if we meet a stop codon, then this process will end. The ribosome will be it will fall apart, and this amino acid chain, this protein, is released into the body. And that's it for translation. And it's pretty simple if you just look at the pictures and just try to visualize it. Okay, good luck on the test. Fighting.